Okay, so welcome back to this afternoon session. Alain Fischer was kind enough to accept to replace Yasmine Belkaïd at the last moment. He won't be able to replace her under all aspects, but he will show us a very interesting story using genetic immune deficits in children as a way to study redundancy within the immune system, and that's as much as I can say about his talk, which I haven't heard before. So thank you very much, Alain, for doing this.
Thank you, Philippe. So I'm sorry, I am not Yasmine Belkaïd. So I will do my best, but I'm absolutely unable to talk about what she has beautifully demonstrated about the immunopathology related to chronic infection. So I will stick to what I know a little bit, which consists in the study of monogenic disorders of the immune system in humans, and to try to take advantage of these many monogenic diseases to try to see whether we can address the question about what is essential and/or, this is the same, what is redundant among the many immunological pathways involved in fighting of infections in humans. So today, as it was 2 months ago or 1 month ago, doesn't matter, we know 338 genes that have been found to be mutated in a context of disease. So these are not variants, these are not polymorphisms, and diseases that are for most inherited with Mendelian inheritance. So the burden of the mutation is quite heavy. So 338 genes causing 346 related diseases. There are more diseases than genes because in some occasions both loss of function and gain of function mutations have been reported. Most of them, and this will be most of my talk will deal with, relates to vulnerability to infections. As you will see, it can be very narrow or in other occasions broad, but we should not forget, and I will mention it in a few occasions, that these inherited disorders are also causing immunopathology, allergy, inflammation, and autoimmunity. And in principle, all aspects, all arms of immunity can be impaired in these diseases. So the questions again is, can we learn something by a broad overview of these 300x genetic diseases observed in humans? I will essentially tell you about 2 stories. One relates with cells, and the second one about genes. The cells relates about the study of the question as written here. Could we determine whether NK cells and innate lymphoid cells, and you may know that they belong to the same pathway of innate cells that are involved in the development of several arms of immune responses that are copying T cells, but they are not T cells, they are not involved in adaptive immunity. So are these cells essential or redundant? And this study was performed in collaboration with Eric Vivier in Marseille, and this was based on a very unique setting in humans where there is a situation, I will describe to you in a second, where patients, so individuals, have no NK cells, no innate lymphoid cells, whereas all other cells, as far as we can tell, all other cells of the immune system are normally present. So it starts with a genetic defect or genetic defects, there are 2 of them, in either the gene encoding the common gamma chain, which is shared by several cytokine receptors, such as receptors for interleukin 7 and 15, depicted here in a simplistic view. And the second genetic defect is the defect in the kinase JAK3, Janus-associated kinase 3, which is related to signaling downstream of this receptor, inducing phosphorylation of STAT3 and STAT5 molecules involved in signal transduction and programming of gene transcription. So in patients who have null or profound mutations in either the gene encoding gamma C or JAK3, they have a profound block in T cell development as well as in NK and ILC cells. So of course, together looking at these patients, it doesn't tell us about what is important in between the T cells and the NK ILCs in terms of susceptibility to infections. Indeed, those patients lack T cells, this is here, they lack NK cells, and they lack the different ILC 1, 2, and 3 as appropriately stained with relevant antibodies. This is an example of the blood of a patient with a severe combined immunodeficiency. This is the disease caused by JAK3 mutation compared to a control. So again, this is an observation, but it doesn't mean much per itself. But those patients they are treated by allogenic bone marrow transplantation. So, but in a usually in a very specific manner, that is, they are not receiving any chemotherapy, any myeloablation, so that their host hematopoiesis, which is simplified here, and it stands as, they have stem cells, they make B cells, they make the different myeloid cell lineages, but they don't make T and NK cells. Once transplanted, there is no engraftment of donor stem cells because there is no empty niche in the bone marrow of these individuals in the context of an absence of myeloablation, but still they develop at least T cells. And this is very long-lasting for more than 20, 30, 40 years. It's possible to detect donor T cells in the blood of these patients. So the question here is, and perhaps there is a direct migration of precursors of T cells into the thymus. We don't know about that. The question here is, what about NK cells? What about innate lymphoid cells in those individuals who were transplanted many years ago? And if there is a deficiency in these cells, is there a phenotype associated with the deficiency in NK as well as ILCs? So they indeed have a deficiency in NK and ILCs. This is the picture of detection of T cells, B cells, NK cells, ILC1, ILC2, and ILC3 in the blood of individuals who are still children, which means they have been transplanted, say about 10 years ago, knowing that these patients are transplanting early in life, during the first year of life, because in the absence of T cells, they can't survive. So these individuals, these are the median colon on each of the panel. They have T cells as compared to control on the left-hand side, as well as compared to another control, which is a group of patients who received an allogenic bone marrow transplantation with myeloablation, so where all of the stem cells are of donor origin. So they have T cells, they have B cells, but you see that in the SCID group of patients, they have virtually no NK cells compared to both of the controls, and there are very few, if any, ILC1, ILC2, and ILC3. And the same observation was made in individuals who were transplanted 20, 30, 40 years ago, who are now adults. They have T cells in normal counts compared to controls. In the blood, they have B cells. They don't have NK cells. They have very few, if any, again, ILC1, ILC2, and ILC3. So indeed, it looks like these individuals have a selective deficiency in NK cells, ILC1, ILC2, and ILC3, that persists following the allogenic stem cell transplantation. But of course, these cells, ILC1, 2, and 3 at least, are mostly localized in tissues. They are circulating in small numbers in the blood. So what about tissues? It was possible to study a few tissues from such individuals based on biopsies that were taken at some time points because of clinical problems. And here you have the staining for T cells in red and in brown for one of the markers of some of the ILCs here in the duodenum, the skin, and the colon, respectively, of 3 individuals who had gamma C deficiency and were transplanted, as you can read, 10 years, 18 years, or 5.5 years post-transplantation ago. So 5 to 18 years ago, they were transplanted. So you can see that they have T cells stained in red, but you can't see actually specifically stained NKp46 cells in these biopsies, neither in the duodenum, the skin, nor the colon. In contrast, these are positive controls of patients who have other immunodeficiencies, RAG1 or RAG2 deficiency, and who have, prior to transplantation, NK and ILCs. And in this case, you can see many brown cells in the skin, the duodenum, or the colon. And this is another kind of staining for ILC2 cells, since they were not revealed by the previous marker, using the ICOS molecule that is expressed by this ILC subset. You can see on the left-hand side panels, both in the colon and the skin, there are very few green cells, a few in the colon, 0 in the skin, in a patient who was transplanted several years ago for such an immunodeficiency, in contrast to positive controls of other SCID patients who have a number of these ILC2 plus cells in colon and the skin. So indeed, there is a cohort of individuals who have now been studied, about approximately 20 of them up to now, with a median follow-up post-transplantation of 20 years, extremes from 7 to 79. So the question, okay, they have a selective deficiency in NK cells, as well as in ILC1, 2, and 3. Are there phenotypic consequences of the absence of these cells? And the answer is no. If you compare their capacity of dealing with infections, including viruses like Epstein-Barr virus and other viruses of the herpes group, they have not developed more infections than other groups of patients who have been transplanted in other settings, which of course suggests that these cells are redundant in function in the context where these individuals live, which I think is an interesting, pretty unexpected observation. And of course, the suggestion, which is presently under consideration, is whether the T cells that are developing the same functions as ILCs are compensating for the absence of these cells. By the way, there is another interesting finding. NK cells, possibly ALCs, are supposed to be involved in the fetus tolerance by the mother. And actually, among these 18 individuals, there are 2 women who were JAK3 deficient and had children, despite the fact they don't have NK cells detectable, at least in their blood. We don't know in the uterus, of course, and have very few, if any, ILCs. So, which suggests that, again, here, for this potential function of these cells in the maintenance of fecundity, it may be a redundant function. So this is about cells. Now I'd like to discuss genes. Based on studies, including one from Luis, the one he mentioned earlier during his presentation. By we, I mean Antonio Rossell, who is in the audience, and myself, we estimated that the number of genes in the human genome that are associated, the products of which are associated with immune functions, is around the number of 1889. Of course, these figures will have to be revisited in the future. If one tries to classify the functions with which these genes or the gene products are associated, most of them are associated with innate immune functions, more than 1,500 of them. A minority, about 1/4, associated with adaptive immunity. Of course, some of them, and maybe more that was shown here, are functions in both innate and adaptive immunity, 200 of them. So here the question is, we have these 1,900 genes. How are these 338 genes associated with primary immunodeficiencies, meaning that mutations of such genes cause primary immunodeficiency diseases? How are they distributed? Is it a random distribution, which would suggest that everything is equal in terms of importance in fighting infections or protecting against immunopathology, or is there a non-random distribution? And the answer to that question here, considering only vulnerability to infections, so not all of the diseases are considered, but most of them, you can see that among the 543 genes that are associated with adaptive immunity, 189, about 30% or so, a bit more as you will see, have been found mutated in association with susceptibility to infections, so quite a number of them. In contrast, among the 1,551 genes, the functions of which are associated with innate immunity, only 59 have been found mutated so far. Of course, the study is by far not ended. There will be more gene mutations found in the future, but this is interesting. I will come back to the distinction between immunity related to neutrophils and everything else in a second. Put them in column just to make it more impressive. Among the adaptive immune genes, more than 1/3 of them have been found mutated, and this is likely extremely underestimated because today there are still many, many individuals who have hypogammaglobulinemias, namely a defect in B-cell immunity, which is adaptive immunity, and those individuals we don't know yet what is the gene and the molecules that are abnormal. So likely, if such a study will be repeated, say, in 5 years from now, my guess is that instead of 34.8%, we will be up to X%, which will be much higher. For innate immunity, it's only 3.9% of genes that have been found mutated. Interestingly enough, you take all of the genes of the genome, and we look what is the frequency of genes that have been associated with inherited diseases, is 15%. So here we are much higher, and here much lower. And the obvious observation here is to say that the distribution of mutations in primary immunodeficiency genes is not randomly distributed. So the immune system in terms of genetics is not equal. and the difference is a factor of 9 between adaptive immunity and innate immunity. If we go one step further, looking not only at frequency but also characteristics of these genetic disorders, we'll see a few more differences. Now, considering first primary immunodeficiencies causing vulnerability to infections and related to adaptive immunity, so 189, 106 are mostly T-cell deficiencies, 54 B-cell immunodeficiencies, 12 deal with antigen presentation to T-cells, and another 17 with effector functions from T-cells inducing innate immunity. So what about the characteristics of these diseases? The phenotype, that is the vulnerability to infections, is permanent. So all over the life of these individuals, they are prone to infections. The spectrum of infections is broad, namely many different classes of bacteria, fungi, viruses, and so on. So the spectrum of infections is broad in virtually all of them, and the penetrance of the disease is high in all of them. All right? Now, if we look at innate immune defects affecting neutrophils and macrophages, essentially defect in cell production, defect in adhesion, defect in the production of reactive oxygen species in phagolysosomes in which bacteria and or fungi have been engulfed, the characteristics are more or less the same. There are not so many of them, 33, but again, vulnerability to infections is permanent all over life, spectrum of infections is broad, and penetrance is high. Now we are left with a small number of diseases that are primary immunodeficiencies related to the innate immunity, but innate immunity that is not specific to hematopoietic cells. So mostly two types of immunity, the one related to complement for some of the complement proteins, and the one related to the innate immune receptors, so the pattern recognition receptors, and the many molecules downstream of these pattern recognition receptors, including, for instance, the many interferon-stimulating genes. So here, first observation already made, there are not so many of these diseases. And then the characteristics of these infections, of the vulnerability to infections differs, for instance, from the one of phagocytic cells, since the phenotype is permanent in only half of them, and these are complement deficiencies. The few immunodeficiencies I will mention on the next slide that are affecting, for instance, toll-like receptor and toll-like receptor pathways are inducing a transient vulnerability to infections in life. The spectrum of infections is very narrow, as you can see, and again, the penetrance is not as high, is only high in 50% of them, and again, this is in the complement deficiency, not in the deficiencies affecting pattern recognition receptors and the downstream pathways. So here, not only there is a difference in frequency, but there is also a difference in intensity of the phenotype observed as far as infections are concerned. So what are the situations, the few settings in which non-hematopoietic specific innate immune responses are non-redundant? There are a few. These are essentially genetic defects that have been characterized by Jean-Laurent Casanova and his group. Essentially, they consist in settings where there is a need, quote, a need, for rapid defense at key places at the interface between the body and the outside world, in a context where there is not yet adaptive immune responses, especially in young children. So, for instance, patients who have mutations abolishing expression and function of IRAK4 or MID88 that are important molecules for toll-like, to most of the toll-like receptor signaling, are associated with invasive infections with Streptococcus pneumoniae. But if these individuals are surviving above the age of 5 or 7 years, then they become asymptomatic. They are no longer susceptible to infections, suggesting that once adaptive immunity has developed, antibody production against Streptococcus pneumoniae, for instance, these molecules become redundant. Another setting is with critical organs. Maybe the most striking one relates to the occurrence of encephalitis caused by herpes simplex virus 1, which has been found associated with several genetic defects, all focusing on TLR3 and downstream molecules involved in signaling, leading to production of type 1 interferon locally in the brain. And these are molecules like TRIF, TRAP3, and a few others. But again, this is quite interesting since the phenotype of infection is extremely narrow, since it also only deals with HSV1 and HSV1 in the brain. not anywhere else in the body. And in addition, this susceptibility is transient in life. So the conclusion from these observations, as far as from what do we know about genetic lesions of the immune system in humans, is that innate immune pathways, those related to pattern recognition receptors, may exert critical roles in immunity, but that may be spatially and temporally restricted, indicating that redundancy is not an all or nothing concept. For instance, we may consider that TLR3 is redundant in terms of immunity to many microbes, many viruses, since this is a receptor for nucleic acids, with the exception of HSV1 in the brain in young children. All right. Of course, I have to add a word of caution about these conclusions, since almost all of these observations were made in the context of quote-unquote modern medicine, that is in the Western world, where the ecology of microbes to which individuals are exposed is not the same as it could be, for instance, in Central Africa. But still, in that context, those individuals, of course, by definition, they have a gut microbiota that they can control. They are exposed to many pathogens, such as Streptococcus pneumoniae, I mentioned, Staphylococcus aureus, many, many viruses. And at least for those groups and many others pathogens, what I said, what I'm suggesting about redundancy probably holds correctly. Of course, we can't exclude that individuals who may have, say, mutations of TLR3 may be susceptible to other pathogens if exposed in other areas to parasitic diseases, Ebola, and so on. So, of course, this concept of redundancy is relative and has something to do with time, geography, and medicine. There are a few other data, notably generated by Antonio Rossell, that are supporting this concept of redundancy. If you look at in databases now of healthy individuals, no longer talking about patients with primary immunodeficiencies, for instance, if you look in the exact database, one finds, and the data are here, that adaptive immunity genes that are associated with primary immunodeficiencies are significantly in frequency in the normal population, depleted of heterozygous protein truncating variants as compared to a neutral model, and more importantly, to innate immunity genes. This is shown here, where the frequency of genes intolerant to heterozygous truncation is only 18% for innate immunity, whereas it's 28% for adaptive, and this is quite significant. So the data suggesting redundancy do not come all from the study of patients with primary immunodeficiencies. And in addition, there are certain innate immune gene products that have been described, not in all, but in most of the cases, in the absence of infections. Dectin-1 has been described with invasive fungal infections, but in many other people who are asymptomatic. CD1A, which is a molecule involved in triggering innate immunity of lymphoid cells, has been found defective without harm. Interestingly, TLR7, 8, 9, TRAP, and possibly IRAC1, important molecule in toll-like receptors and pathways, have been found absent in individuals without any susceptibility to infections. So all of these data converge with the concept that these innate immunity pathways are, at least in part, redundant. Now, if one looks at immunopathology and no longer at infections, the picture is quite different. Let's consider, I will not go into too many details, but I will discuss with you three kinds of cytokine-triggered pathways. The first one is type 1 interferon, and back to genetic lesions, genetic diseases in humans. We know only 4 diseases with a very, very restricted, as I said, a narrow repertoire of infections associated with defects in responses to interferon type 1, such as IRF7 and IRF3. And by the way, very few genes and very few patients, not less than 10 patients worldwide, extremely rare. In contrast, we know 20 monogenic diseases that are characterized by an excess of production of type 1 interferon. These are the so-called Aicardi-Gouttières syndrome and associated with inflammation in the brain as well as autoimmunity, lupus-like. So there is quite an imbalance between gain of function and loss of function lesions in terms of frequency for the interferon type 1 pathway. Same for IL-1. Today, up to now, not a single genetic defect in IL-1 production in terms of loss of IL-1 production has been described, whereas we know 10 genetic diseases where there is an excess of IL-1 production. One of them has been discussed today, which is mevalonate kinase deficiency. And these are associated with so-called AIS, which stands for autoinflammatory syndromes. Now, if we look to cytokines, I will only show you one, but there is at least a second one, which is gamma interferon. But just concerning IL-17, which is, as you know, a cytokine or group of cytokines associated with Th17 responses, we have the mirror image. There are 9 conditions, 9 different genes that have been found mutated in which IL-17 production or IL-17 signaling, induced signaling, is defective. And they are all associated to susceptibility to fungal infections plus additional infections, bacterial and so on. Whereas there is only one genetic disease where there is a gain of function mutation in a gene causing hyperproduction of IL-17, which is gain of function of STAT3. So you see this mirror image, which suggests now I'm being more and more speculative from now up to the end of my talk. But just at least for the sake of discussion, it suggests that gain of function of innate immunity is much more frequent compared to loss of function, suggesting that regulation of innate immunity pathways may be more permissive. All right? In contrast, for adaptive immunity, there are, as you may know, multiple layers by which it is put under control, which starts from during differentiation of cells, where there is deletion of high-affinity self-reactive T and B cells to avoid autoimmunity, gene editing in the periphery, regulatory T cells. I'm not going into too many detail. Gene editing, self-antigen-specific T and B cells can die because of the interaction between the Fas ligand and Fas molecule, and there are many regulatory molecules that are acting in cis at the surface of T cells and sometimes B cells or in the cytoplasms, such as CTLA4, LRBA, and ubiquitination pathways regulating signaling by the T cell receptor and the B cell receptor. Interestingly enough, all of the molecules that are listed here have been found mutated in patients who develop autoimmunity and or inflammation, indicating that they are all essential. Nothing is redundant here among these many molecules, suggesting again that adaptive immunity is strictly regulated, and if one pathway is missing, then it is leading to major immunopathology. In contrast, in the field of innate immunity, especially immunity of pattern recognition receptors and their pathways of signaling, there are only 4 defects known so far in these molecules. I will not go into the detail, which suggests again that maybe defects in regulatory pathways of innate immunity may be more permissive in terms of leading to inflammation. So perhaps this is extremely speculative, but I would suggest that for innate immunity, the circles are not cells, they are pathways. There are many, many effector pathways, that's for sure. This is pretty well known. But perhaps not so many critical regulatory pathways. There are several, they have been described in details. But many, not so many, are absolutely essential, suggesting that these multiple effector pathways, become of evolutionary pressure made by many pathogens, are not so intensively regulated with the overall, of course, it's very much simplification, but there is overall a balance shifted towards host defense, that is effector functions in comparison to regulation. For adaptive immunity, the balance may be different. This is clear. The effector pathways are less numerous compared to innate immunity, but in proportion, regulatory pathways look to be more important, maybe more numerous, at least more critical, since every single mutation of the molecules involved have been found to cause diseases, autoimmune or inflammatory diseases in humans. So suggesting that these pathways are much more intensively regulated, and the balance here is much more equilibrated, if I may say so. To finish with, I'd like to discuss these findings in the context of cost of immune defenses. This has been already alluded to in several of the talks today. An immune response against a pathogen or even against a non-pathogenic microbe has a cost in terms of direct energy to provide the effective functions for cell division, which is very important, for instance, for T cells that expand tremendously at the time of an immune response. And there is the indirect cost, disease prevention, as we heard, control of immunopathology, repair of lesions associated with infections. And interestingly enough, we found that there is an inverse correlation between the cost of an immune response, as proposed by Ruslan Metsitov in a review a couple of years ago. I have slightly modified, and there is an inverse correlation between the cost of an immune response and the redundancy of the genetic disorders in humans. So let's describe it in a bit more details. If we go about the cost, what is the less costly among immune responses are everything which is constitutive, which is mostly neutralizing microbes and not involved in most of the cases in inducing cell lesions and requirement for repair. These are the immunity related to microbial peptides, as well as immunity related to secretory IgA and IgM. Interestingly enough, for instance, IgA deficiency in humans is known, it's pretty frequent, but in most of the cases it's redundant. So most of the individuals who have no IgA have no susceptibility to infections. So the redundancy of such potential genetic defects, and so far not a single defect in antimicrobial peptide has been described in humans, despite the many, many genes encoding such peptides. So then if we go to pattern recognition receptors and activated cells, epithelial cells and many others, the response here may not be that costly because it is not associated with cell proliferation, for instance. It's not causing that much lesions and inflammation. And again, as I showed you, these pathways, as numerous as they are mostly redundant. And now I will not go into all of them, but NK and ILCs, again, there are very few of them, so that likely the cost of using them in an immune response may be not that high, and I showed you that there is evidence for their redundancy. In contrast, if you take cytotoxic T cells, Th1, Th2, Th17 B cells, their involvement in an immune response as well as neutrophils is very high because there is a lot of cell division, including the production of neutrophils every day in the body. And they are producing a number of toxic molecules that are causing cell death and immunopathology. And those are the pathways in which redundancy in humans, as observed from immunodeficiencies and a few other studies, looks to be the immunodeficiencies are present, so there is very little redundancy. So to me, it's pretty striking to see this inverse correlation, which may suggest the following, that during evolution, because of the selection by a number of microbes during human and even, of course, pre-human evolution, there has been, by duplication, the generation of many, many constitutive pathways and as well as innate immune pathways, mostly related to pattern recognition receptors that have been developed, but their involvement in immune responses is not that costly. In contrast, to use neutrophils and/or T cells and B cells in an immune response is going to cause lots of damages that cannot be fully accounted by an individual, so that in terms of equilibrium, they had to be selected in a balanced manner to limit their function so that there are not so many genes, not so many pathways, and they're heavily regulated. Of course, it's just a model. So I will finish here just to propose that there is some redundancy in the human immune system, at least as studied in the context of the Western world. This should not be forgotten. But in any case, it's not an all-or-nothing concept. It's also tissue and time-dependent. Of course, the study of these monogenic diseases in humans are pretty interesting to study the importance of relative pathways, despite obvious limitations. And in terms of evolution, we may propose that evolution as favored in the context of selection by the many pathogens with which we have been living with, have favored the multiplicity of defense tools that have a relatively low cost effectiveness ratio. Incidentally, practically speaking, in medicine, if what I'm proposed is correct, it suggests that if each time there is the choice, the possibility to treat an immunopathological disease, it would be better to use, to target immune pathways that are essentially redundant to avoid infections. For instance, if you, as it is done, if you neutralize IL-1, as it is done, for instance, in inflammatory bowel disease, the risk of infection will be reduced since the IL-1 pathway looks to be apparently redundant in infectious immunity compared to neutralizing IL-17 or gamma interferon. For cells, it would be, if, of course, this is accurate, better to neutralize or to kill or whatever, to block NK and IL-6s that look to be essentially redundant versus the T cells that are absolutely critical. So this may help a little bit to decipher strategy of immunosuppression in the context of autoimmunity or inflammation. I will stop there, just mentioning a few names of people involved in these studies. The work on the NK cells and IL-6s in the context of severe combined immunodeficiencies has been carried out together with the group of Eric Vivier and these people, also at Imagine with Bénédicte Neven, Capucine Picard, and Julie Bruneau, and the study of the redundancy has been done in close collaboration with Antonio Rossell. Thank you very much.